Okay, let's go ahead and get started. Good afternoon, good morning, and good evening to everyone who is attending wherever you might be. I am Rob Burgess, and I'm Chief Business Officer of Sino Biological, and I would like to officially welcome all of the attendees and most importantly, our very honored speaker, Dr. Rob Durham of Gyros Protein Technologies, to the next installment in the Sino Biological hosted and sponsored series on COVID-19 immunodiagnostics. Thank you all for being here today. I want to make a few announcements regarding housekeeping issues. All of the attendees here today will be muted throughout the seminar, and after the seminar, you cannot ask questions verbally, but you can ask them in the chat box, which is provided. So I would ask each individual to think of some good questions for Dr. Durham and type them into the chat box. And after the end of the seminar, I will select questions from the chat box and relay them to Dr. Durham for live answering. In addition, all the attendees that registered and attended will get a link to a recorded version of this webinar very shortly afterwards. So with that, it is my pleasure and my honor to introduce to everyone today our keynote speaker, Dr. Rob Durham, whose title is the Director of Service and Scientific Support for Gyros Protein Technologies. A little bit about Rob. He's been with the company for 10 years now, but prior to joining Gyros, he was actually a customer of Gyros as a principal scientist in translational medicine at Pfizer. Rob received his doctorate in pharmacology and neuroscience from Michigan State University, and he completed his postdoctoral fellowship training at Park Davis Pharmaceuticals before making his move to Pfizer. So the title of Dr. Durham's talk is Microfluidic Nanoliter-scale Immunoassays for Rapid SARS-CoV-2 Antibody Analysis in serology testing and vaccine and biotherapeutics development. So Dr. Durham, we're very happy to hear from you today, and you can go ahead and share your screen.
Great, thank you so much, Dr. Burgess. I really appreciate the opportunity to be here today. And the title is quite a mouthful, so I appreciate you getting through that. Go ahead, share my screen. There we go. There we go. I think that's it. What I'd like to do today is talk a bit about the Gyrolab platform, how it's used, and then specifically how it's used in the COVID-2 assay for serology, as well as some other applications related to vaccine development. So our nanoliter-scale immunoassays are designed for time-critical workflows, and what this is a robotic system that uses a unique CD-shaped assay plate to allow you to automate and miniaturize immunoassays. So if we start over here on the left hand, you can see the real heart of this technology. This is a streptavidin-coated bead particle that we can use to functionalize with any biotinylated capture reagent, and in conjunction with an Elexifluor or some other fluorescently tagged detection reagent, create a sandwich ELISA for analytes of interest, particular interest to the bioanalytical community. What's unique about this system is that bead is incorporated into a 15 nanoliter affinity capture column. This tiny 15 nanoliter column is built within a microstructure that is the well on our CD-shaped assay plate. So there's one microstructure, one data point, and I'll talk more about how this all works together, but suffice it to say, these channels and grooves allow us to deliver reagents and samples, measures them, and delivers them in an automated way so that we can miniaturize and automate an ELISA type assay. Our CDs are organized in segments of 8 microstructures connected by a single common channel. We have several different CD types that we'll talk about. The principal difference between them is the size of this volume definition sample chamber here, which measures the sample volume and then delivers it to the microstructure when the CD is spun. Now, so because the sample volume definition chamber determines the sample volume, we have 1,000, 200, or 20 nanoliter CDs. So that sample volume chamber is the defining characteristic of each of those CDs. So what this does is that means that on CDs with 1,000 nanoliter volume definition chamber, we're going to have only 12 segments per CD because the real estate taken up by that sample volume definition chamber is a little larger, versus a 200 or the 20 nanoliter CD where we can squeeze 14 segments of 8 microstructures on there. So that's the difference between 12 to 14 segments per CD. When we read the CD, we actually see an image, a three-dimensional image of the column profile, that affinity capture column, where things are binding on the column, and the retained fluorescence on the column is then a measure of how much of the specific analyte was there. So the integrated area underneath that fluorescence peak is then interpolated into converted into a response, which can then be converted to a standard curve, and because we're using fluorescence and a flow-through design, we get very broad dynamic ranges. I think some of this is a little easier to see if I show a little video. So here's a video using a 200 nanoliter CD example. Again, it's showing, oops, let's go back. I hit the start button there. Okay. So now what you're seeing is capillary action is drawing the capture reagent into a segment. Those hydrophobic barriers are stopping capillary flow at strategic locations, so that now we've trapped 200 nanoliters of capture material above each column. And then when we spin the column, centripetal force delivers that capture reagent to the column and functionalizes the streptavidin coated bead with our biotinylated capture material. Now, for brevity, I haven't included wash steps that happen in between each of these actual steps of the assay that would rinse off any unbound material. Next, you can see the liquid handler is delivering the samples. Again, we're not doing any manual pipetting here. The liquid handler does all the work. It delivers the sample. It actually overfills it slightly. This is a volume definition chamber, and then we spin off the excess so we can precisely define that 200 nanoliters of sample that is then spun at a higher speed and delivered to the column. And in flows over the column at about 1 nanoliter per second. Next, the common channel is used again to deliver the detection reagent. Our fluorescent tag detection reagent is divided up, and then when the CD is spun, 200 nanoliters is applied to each of the columns simultaneously. Again, because this is a disk and everything is happening in flow, it's all happening in parallel on the CD, so we have no time-dependent lag in between any sample receiving the sample or the reagents. Next, you can see as the CD spins, we actually scan the columns with a laser and it rasterizes down that column. And what you get is that three-dimensional image of the retained fluorescence on the column, which is then converted into a response. So again, this microfluidic design really eliminates pipetting errors. We're using physics to define the sample volumes and control the flow over the CD. Individual nanoliter volumes are precisely defined because of that volumetric space on the CD can be created and manufactured very precisely. And all the samples are processed in parallel. So our mission has really been to transform standard old-fashioned immunoassays, which have been the standard way of doing business since the 70s when the ELISA was introduced, to transform that by enhancing performance and productivity. So by performance, we want to have a robust platform with a very broad dynamic range. High precision and reproducibility is owed to the automation, and it's suitable for a variety of matrices because everything is happening in flow. On the productivity side, because we don't have any long incubation periods, we can get really fast assay turnaround. It takes about an hour for samples to run start to finish on the system in a completely unattended fashion. And that fast turnaround time means that you can have data to drive your decision making rather than making decisions at risk and waiting for the data to come in later. This also saves analysts time. Increased throughput and reduced repeat analysis means that this is an easy technology to use in places where you have to transfer assays from place to place. If it's a manual ELISA, there's a lot of art involved in doing executing a manual ELISA, and so taking the art out of the assay and automating it really enhances the productivity. We offer, in addition to the Gyrolab instruments and the consumables, the CD, a host of ready-to-use kit and various softwares which can add to the functionality and utility of the Gyrolab platform. And I won't spend a lot of time here, but if you're interested in other applications, other immunoassay applications, Gyros is used in a wide variety of places. Anywhere an ELISA is typically used today, the Gyros is a potential fit. So what I'd like to talk about first is a Gyrolab immunoassay for detection of anti-SARS-CoV-2 antibodies in serum, a serological assay for the detection of anti-SARS antibodies. So I'm sure, as many of you know or have learned in the recent months, the receptor binding domain, or the spike protein, is an active target for vaccine and therapeutics development. This S1 antigen is shown to best distinguish the COVID-19 patients from controls or other other COVID-related virus particles. The Gyrolab assay is a qualitative detection of total antibodies generated against the spike protein domain of the SARS-CoV-2. How this works is it's a bridging format. So in our system, the way this works is you have your biotinylated RBD protein, receptor binding domain protein, as a capture reagent. And then we have the SARS-CoV-2 RBD antibody, or some positive control, is the blue antibody-shaped molecule. And then we have a detection molecule, which is a receptor binding domain with FC that's been Alexaflor labeled. So what this allows us to do is to develop a bridging assay on our beads that can be used in our automated system. So the positive control is a polyclonal antibody that's raised against the SARS-CoV-2 receptor binding domain, and it's research application and preclinical studies. It's also clinical applications. It could be an assay that's suitable for CLIA certification. So what we've done is taken reagents from Sino Biologicals and applied these to the Gyros so that we could create this assay. So just for specifics here, so that anybody could obtain these reagents and build the same assay in your own lab, I've included this table which shows the spike protein and the catalog numbers we use for the capture reagent, the labeled reagent for the detection molecule, and the spike positive control in this case is a polyclonal antibody, all from Sino Biologicals. We're using in this assay the BioAffy 200 nanoliter CD, so that means you could get up to 112 data points in a single CD, so 14 segments of those 8 microstructure segments on a single CD. So response against positive control samples. So we've got a negative control sample where we see what the baseline is, and then we've done a serial dilution of our positive control sample out to from 40 nanograms all the way out to 200 micrograms per mil, and you can sort of see the responses there. Nice response curve. And this is all done in 25% human serum, so that gives us a sense of matrix tolerance and so forth. So all acceptable matrix tolerance, very nice reproducibility. Specificity, so we want to be sure that if we spike back some positive control that's been spiked back with non-labeled receptor binding domain, then we should be able to specifically reduce that signal back to 0. And that's in fact what we do. So here at various concentrations, see at 0.25, 2.5, and 25 micrograms per mil, this is the concentration of the additional unlabeled RBD material in the presence of a positive control at 1,000 nanograms per mil. You can see that we can start at 90% inhibition and inhibit that signal all the way back down to 0. So very good specificity. This is a group of 50 pre-pandemic individuals showing control level, no cross-reactivity with any other antibodies in serum. And we have several negative controls in addition to some positive controls, again showing that we're getting good specificity between samples and positives. Negative samples and positives. So the Gyrolab total antibody assay for the SARS-CoV-2 is established and tested in positive controls and pre-pandemic serum. The assay was successfully distinguished between positive and negative samples. While I don't have data that I'm able to share at this time, we did test some real samples and find we had good specificity in that sample set. I'm sure that will come out at a later time, just not able to share the data today, but it successfully distinguished between positive and negative samples, and from good sensitivity down to about 50 nanograms per mil of our positive control. We did a 1:4 dilution of samples, so we're still working in about 25% matrix. The microfluidic assay format allowed us to consume very little of the total sample. So even though in human subjects you're likely to have not be volume limited, certainly in preclinical species, rats and mice, so forth, you are very limited in sample. But even in human studies, I've known situations where you want to get the most out of every one of those precious samples, and so low sample consumption is certainly something to think about. But what's really useful is we're really dramatically reducing the amount of material required to run the assay. So that 100 micrograms of the receptor binding domain his-tag material is enough to do about 120 CDs or 13,000 data points. So you're going to stretch your materials a lot further if you use a Gyrolab platform. So again, looking at low reagent consumption, low sample volume, quick analysis time, about an hour to do the 56 samples in duplicate, good sensitivity, and very broad dynamic range from 50 out to 200 micrograms per mil. So that's kind of an introduction to the serology assay that we've developed using the Sino reagents, and I think it's a really good start, interesting application. Is something that anybody out there with a Gyrolab who's interested could use this information to apply to their own research. I'd like to take a little bit of time and talk a bit about how the Gyrolab has been used in vaccine development to enable faster decision-making, real-time data analysis. So again, the Gyros is used in vaccine workflows anywhere you would use a sort of ELISA assay today. So detection of antigen-specific antibodies in serum, or antibody affinity, or reagent screening, all those places. Or if you have to do a host cell protein, measure contaminant proteins, things like that in the purification process, you can again utilize a Gyros assay to do that, automate it, and miniaturize it. So it's very commonly used in serum and plasma, and formulations with aluminum particles or other formulations in various emulsions, which can create some problems in a typical ELISA assay. We have very good matrix tolerance in the Gyro system, so it helps to reduce the problems that you might encounter in a typical static ELISA where matrix can have a big impact. What I like to do is do a couple of case studies to sort of put this a bit in perspective. So the first is detection of antigen-specific antibodies in serum. So it's not unlike what we just reviewed now. What we want to talk about is how you could test various adjuvants, and you could have various different isotypes of antibodies you could be interested in measuring. So in this experiment here, we're looking at 3 different adjuvants. This is all done in mice immunized with the Neisseria antigen and different adjuvants. And what we wanted to do here is to look at various isotypes of antibodies that were formed over time in these mouse samples. And you can see the samples were collected, and we actually did several different assays using our small sample volume requirement. We're able to get several different assays out of each of these samples. Now again, these were all done where pools of sera from 10 vaccinated mice were initially done in this particular experiment, but it allows us to look at different antibodies that may be pulled out of the system. So we saw several different subclasses of antibodies, and it helps to understand which adjuvants are providing the best, most potent vaccine development. So the platform really reduced the sample volume requirements, allows you to screen multiple IgG subtypes in a single sample. So 8 microliters per sample per assay. The gyro system also gives us an advantage over a plate-based system because we're really reducing the sample reagent volume and minimal hands-on time. We're getting our data back in about an hour, and we can get capture of specific IgGs in a wider dynamic range than you would typically get in an ELISA. So that broad dynamic range means you'll have to do fewer dilutions of your samples to hit your target. The next one is talking a bit about potency testing. Vaccine QCs, mouse immunogenicity used to test batch-to-batch consistency and vax potency and stability studies. Fermentation and purification process validation optimization, and so all about producing the vaccines and comparability studies when they have new lots or new batches of vaccines, or if there's adjuvant or formulation changes, they have to do these quality control checks. So most potency testing procedures in the in-life situation, immunized mouse strains. In this case, they're using BALB/C mice, and then the antigen samples are normalized to a starting dose. So they do a series of 2 to 3-fold dilutions to create 6 to 12 dosing points, and then they do this same sample analysis on the Gyrolab system. So what they're going to do is do an ED50 based on the seroconverted mouse number per dosing group, so that we get about they end up with about 120 to 220 samples to obtain an ED50 value. And then they have to classify these, and the data is used to support the BLA and GMP documentation. So again, the potency pain points are a large number of samples. The Gyrolab assays can do the 112 data points per CD. It can be tedious work, especially if you're doing a manual assay. In this, you load the samples into this plate, and then the instrument automates and runs the samples start to finish. Low sample volume, when we're working with mice, we only have about 8 microliters per sample required on the gyro system, so it really dramatically reduces the number of bleeds and the amount of samples that you'd have to pool together in order to get a sample valid result. Variability in animal studies, the gyros automates and helps reduce the variability on the assay end of things, and so that can help improve performance overall. Then of course our system is 21 CFR Part 11 compliant, so this is completely compatible with being included in IND and BLA filings. So this is just a look at reproducibility. So here we've got 5 different tests. So done with a serial dilution of samples, and you can see an example of the mean response curve over here and the several dilution factors. And then this just gets at what the individual CVs are, the group CV across the group, and you can see that we get really good reproducibility both within runs and then across several different runs. We're seeing acceptable reproducibility with CVs less than 10%. So that's important in order to be able to make a decision about the stability of your assay. Will help to ensure that you're making good decisions about the potency of your drugs as they're coming out of QC testing. This is looking at comparison of vaccine potency comparison of vaccine potency and assay in the Gyrolab and LUMINEX. So we're comparing Gyros versus LUMINEX. And again, this is with a couple different groups of monkeys at different time points. We're looking at the response rate, the positive responses in response to these vaccines in the monkey group here, using the LUMINEX assay, and over here we're doing the same assay on the Gyros. And while you see the response units don't correlate to one another very well, but you do see the same trends, and the same rank order of the dose groups was observed on either the LUMINEX or the Gyros assay. So you wouldn't have made any different decisions about who were the high responders from looking at either the Gyros assay or the LUMINEX assay. So again, the Gyrolab immunoassay supported both mouse and monkey vaccine potency studies. It's fully automated, less hands-on time, and quicker turnaround time. And the sensitivity and reproducibility are certainly comparable to LUMINEX in this study. Last thing I want to talk about is a biotherapeutic PK application. In this case, antibody therapeutics for COVID-19 include things to tamp down the immune response or cytokine storm that may result from exposure to COVID-19. And in this case, an anti-IL-6 antibody, ACTEMRA, is one of the potentials that's used to block the cytokine storm by blocking the IL-6 receptors. So we've put this assay on the Gyros. Again, this is a 3-step bridging assay on the Gyrolab, where we have a biotinylated human IL-6 receptor as the capture molecule. We have the drug itself as the analyte, of course, and then a recombinant anti-human anti-idiotypic antibody is used as the detection molecule in this case. So, so again, we can sandwich our drug in between a capture of the specific target capture, the IL-6, and an anti-id detect that's been labeled with Alexaflor. In this one, this is another sheet that's on our site, so you can get a sense of what can be done with the Gyrolab. We have a wide dynamic range between 160 to 60,000 nanograms per mil in this particular assay. Inter-run precision is less than 20%. The samples here were run in triplicate with 3 runs. Again, it takes less than an hour to do a run on the Gyros, and this sensitive PK assay that would be suitable for both preclinical and clinical studies. The estimated LLOQ in this assay is about 160 nanograms per mil, with an upper limit of quantitation of about 60,000 nanograms per mil. So again, it shows how you can use a flexible, open Gyrolab platform with specific reagents to develop a PK assay in relatively short order. So what I've tried to show today is that the Gyrolab microfluidic system supports serology and immunogenicity for vaccine development and antibody-based therapeutic PK applications. It's been widely used in these areas, and again, I think the reason people adopt our technology is because they get the data back fast. You know, I spent at my time at Pfizer, we worked in a lot of project teams to support them, and I never knew a project team that wanted the data back slower. They always want the data back sooner. So I think speed, especially in today's environment is critical. Oftentimes it can be a game changer. And so being able to get your data back in an hour and that short cycle time means you're going to get a lot of efficient use out of a gyro system. So I hope that gave you some food for thought, and if there are questions, I'm really looking forward to continuing the conversation, and thank you so much for your attention.
Thank you, Dr. Durham, for that excellent talk. I and all the attendees really appreciate that. We're a little bit limited on time, so I'll jump right into the question and answer session.
Okay.
Just all the attendees, you can type your questions into the chat box. It's not too late, and I will relay those to Dr. Durham. So we've already gotten a few excellent questions here, some of them a little more general and some of them a little more technical. I'll start with one that's really more about distinguishing between inactive virus versus maybe an active infection. There's been some controversy about just focusing on the spike protein only and that you may be detecting inactive virus. So I'm wondering, just from your perspective, have you thought at all about targeting other proteins on COVID-19 that are really only expressed during an active infection?
Yeah, I think that's a really excellent question, and certainly more work needs to be done. Where we have focused our efforts is to as a tool maker. You know, I'm not in a position where I have the R&D resources to go after the deep biology or that, and I think that's absolutely an excellent question. What we've tried to do is see, could we apply our technology to this area? And then what we'd really welcome is how would you do that differentiation? And is there some other reagent, other formats of the assay that we could develop in conjunction with somebody to address that question? Certainly we understand, you know, we pulled this assay together very quickly and with great reagents that, you know, we built a really robust assay, I think, but it certainly has limitations. And I think, you know, certainly anybody with a better understanding of the biology of this virus than myself, we could partner with you and show you how the gyros may enable you to do more work, build different assays, different formats that could really enable your work.
Yeah, that's great. And I would encourage those that are attending today that if you have other targets that maybe focus a little more specifically on an actively infecting virus, you might want to reach out to Dr. Durham and others at Gyros and strike up a collaboration.
Yeah, because I think if you know the targets, you know your biology, you know, I can help you use our tool to its greatest advantage. You know, that's the area that I can help out with.
And there's a question about mutation rate. I think COVID-19 has a relatively high mutation rate, at least for some of the proteins it expresses. Did you see any kind of a variance in your actual test results?
Yeah, so with so limited, our test results are so limited right now, and what I actually have seen data from is extremely limited. So I couldn't say, you know, and certainly, you know, certainly that mutation rate is something that I don't fully have a handle on, and how it may affect both infectivity and where those mutations are likely to come. What's more conserved? I wouldn't even have a guess. But what I can say is that with the materials that we've had, we were able to develop a pretty specific assay. One of the things I was worried about is would we be seeing other SARS-related antibodies or other SARS-related antibodies pull down in our assay. And so far, the limited data I've seen is that they're very happy with the specificity of the assay.
Can I ask you, related to that, are you planning to move this into like a CLIA format, or maybe?
Well, I think not us, but certainly, yeah, I think somebody could. You know, I think certainly we've played in the lab developed test space with other companies that use our technology for diagnostics, and so it's definitely something that could be done.
And one quick question, did you experience any positives in the pre-pandemic pools of patient samples you tested?
None that we tested. No, no positives.
Okay. A technical question about the system itself. Could you substitute the fluorophore that you're using with maybe a fluorescent nanoparticle or a quantum dot or something like that?
That's interesting. So our technology uses a monochromatic laser. So it's designed to get those low red dyes. So Alexafluor 647 or Dye-Lite 650. So something in that range. So certainly if you had a particle that was fluorescing in the right range, we would pick up a signal.
Right.
What the advantages of that would be over the standard dye, I'm not totally certain, since what we're really trying to do with our detector is once we've captured the material on the column using the capture reagent, we want our best specificity with the capture reagent. So once it's fixed the material on the column, we really just want to have an even paint the column basically with a low but robust signal, so we can differentiate positive signal from negative very effectively. And oftentimes in that case we'll titrate down to a lower detect concentration to make sure that we can differentiate. We get better separation between a blank, say, and our low QC, our low signal, to help improve that. And so I don't know what the advantage of the nanoparticle necessarily would be
Right. Right.
in our system.
Still a good question, but
Yeah.
I guess the system, like you said, is monochromatic, so there would be no chance to do multiplexing using
Yeah, yeah, we don't have an easy entree to do multiplexing with the laser setup as it is now. We've taken the tack of we miniaturize the sample volume requirement enough so that, you know, what I used it for when I was at Pfizer was to do essentially a multi-sampling experiment where I could take 10 microliters of mouse sample, and I could take that 10 microliters and split it into several aliquots and do 5 different biomarker assays on that sample with minimal dilution of the sample, because I really only needed 4 microliters of sample to get a duplicate measurement. So we do this multi-sampling approach or gyroplexing approach where we can take a sample and split it up in an automated way so that we do individual singleplex assays, and that's kind of been the approach that we've taken so far. But it's a question that comes up quite frequently, and I think as long as if you have an experiment where you wanted to look at 50 different things or 100 different things, well, you're going to use something like LUMINEX as a multiplexing strategy, right? Where they can look at panels of things and get an idea, more of a fishing expedition. But if you can break it down to 3 or 4 or 5 things that are critical to you to measure, features that are critical to measure, the gyros is a good option to do this multi-sampling strategy rather than a true multiplex.
Yeah, I guess in a way it is a multiplex because you're only using, what, 4 microliters or something of a sample, and sample volume is so crucial to save. So if you have a reasonable sample volume, you can split it and just do multiple targets, right?
Yeah. Yeah, I had one of our customers who's pretty brilliant and is always thinking about different ways to do things, wanted to do an isotyping experiment where he said, okay, I'm going to actually put the sample through the common channel rather than in the sample inlet, and then that way it split it into 8 different 200 nanoliter samples, right? And then he put the capture and the detect reagents in through the sample inlet. So in that way, that allowed him the flexibility to take each sample and split it into 8 individual aliquots automatically on the CD, and then multiplex in that way, right?
I guess you save sample and you save consumables that way too, right?
Exactly, right, exactly.
Quick technical question. Somebody is asking you to maybe just explain how the optical detection system is set up, maybe without revealing trade secrets.
Well, it's a laser-induced fluorescence. I don't know. Maybe I'm not the best guy to answer the details on that. I can't give away anything because I don't know enough to give away.
Right.
But basically, so we have our monochromatic laser, which hits the little column of streptavidin-coated beads, and we have a photomultiplier tube that then shoots that signal into a detector. Now we can play around with the gain settings a little bit, and oftentimes we'll scan at 2 or 3 different gain settings when we're doing assay development. But it gives us the ability to get about 4 to 5 logs of instrument dynamic range, and within that we can typically get 3.5 to 4 logs of linear dynamic range out of an assay. So I think it's the setup does avail itself of getting relatively broad dynamic range compared to, say, a colorimetric ELISA assay.
That's great. That's great. And speaking of the instrument, let me just ask you kind of a business question. Can you tell us how many units are currently in the field?
I would say on the order about 250, 300 worldwide.
That's great. And the system for any particular targets has been approved for clinical diagnostics, or I mean, what's the status of that for like non-coronavirus related?
So again, I'm not at liberty to disclose the particular lab, but we do have a diagnostic lab that uses our technology for lab developed tests. So they have esoteric tests that they have approved and used, and they use our instrumentation quite heavily for that purpose.
That's great. That's great. You mentioned LUMINEX. Looks like you're getting some very comparable results to LUMINEX. Can you give us a comparison of the pricing, maybe, of an LUMINEX system versus the Gyros platform?
Yeah, it's been a while since I bought a LUMINEX system fully outfitted, so I don't have a great sense there, but we have 2 base systems. For your local pricing, you really just feel free to reach out to a salesperson, and then you can get what the local pricing is for where you're at.
Great, great.
I can put somebody in touch with a salesperson if they really want to.
Remember that, attendees.
Yes.
Get you some good pricing. Special seminar-based pricing, right?
Yeah, now I'm going to get in big trouble already.
That's right. Pressure's on.
I said the pricing number.
Right. So. The pressure's on now, right?
Yep.
Let's see. We have a few minutes left. I want to try to get to all the excellent questions here. So there's one. Could you give an example of the process of assay development using a custom reagent or target?
Yeah, I can. So it's a multi-stepped approach, but it's pretty straightforward. So what we typically do, my approach is to go into the lab, and we'll have someone will have their capture and their detect reagents that they've selected for their particular assay, and one of the things I encourage people to do is make sure you have the best quality reagents, right? So oftentimes I'll go into a situation where we're evaluating the technology and somebody will say, we could never get this assay to work on an ELISA, so we're going to throw it on your platform and see if you can make it work. And I'll say, well, we're probably not going to make it work because if it's the critical reagents are the problem with your assay on the ELISA, they're likely to be just as big of a problem on our platform. But as I alluded earlier, what you want to do is select the highest specificity, highest affinity reagents as the capture reagents, because then you take advantage of the flow-through design to actually affinity purify your sample over that 15 nanoliter affinity capture column. So select good reagents, your highest affinity to be in the capture orientation, and then we'll do a little simple best pair analysis where we'll take all of your captures and all of your detects and run them against each other. And the nice thing about that is that you can take the same standard curve on that 200 nanoliter CD. That gives you 14 different pairs of combinations you could try. So in singlet, you can do 14 different capture and detect combinations in around that CD, and in 1 hour you do your DOE to establish what's the best pair. From there, we'll do a little bit of optimization in terms of what's the detect concentration. We might want to, as I said before, titrate down that detect concentration to improve the signal to noise. And then do some matrix tolerance tests to see what is the final minimum required dilution we're going to have to deal with in order to make the assay work. And I'll say that because of the flow-through design, again, oftentimes we get away from having to do huge dilutions because of matrix intolerance. You know, the flow-through design means it either sticks or it doesn't, and it gets washed away. And unlike a static ELISA, where you have more time for long-term, slow on-rate sort of interference things that can happen, you have a much better situation there.
Great. And the follow-up to that is just how long does it take to do a typical custom assay development project?
Yeah, I would typically say we'd be able to get an assay up and running in about 3 or 4 days. So compared to a typical ELISA assay, it's really fast. You know, again, one of the things that you get away from is that you don't have to have a blocking step or figure out which plate we're going to use. Again, that sort of flow through design, if you have really good capture reagent, you're going to help make that super efficient in terms of affinity purifying your samples with your capture reagent as part of the assay. Really is a super efficient assay.
3 to 4 days, that's very impressive.
Yeah, again, it's that cycle time, right? So you're going through the same decision tree of experiments that you need to do to develop an assay, but when you have a static ELISA, it takes 5 hours to get start to finish. If you're aggressive, you might get 2 experiments done in a day. On the gyros, you're going to be able to get 3 or 4 or 5 experiments done in a day. That cycle time is what drives the speed with which we can do the assay development.
Yeah, that's fantastic. It looks like the remaining questions are rather technical. I think we'll go ahead and wrap it up now, Dr.
Okay. Yeah, if somebody has a specific question they feel like they wanted to talk a bit more about, you guys have my email address. Please have them reach out to me. I'm happy to have a discussion.
Okay. Sure will. And we'll close the meeting now. Dr. Durham, first I want to thank you for an excellent seminar and your time today. We sincerely appreciate it, and Gyros is a wonderful partner for Sino Biologicals. So thank you all once again for being great partners and collaborators with us.
Good. Our pleasure.
Yeah. And I want to thank all the attendees. We had great attendance today, and thank those who asked excellent questions that were very thought-provoking. And I remind everybody, we still have, I think, 5 more COVID-19 immunodiagnostics webinars coming up. So please check the Sino website and register for those. And I want to thank my colleague, Jen Liu, who has organized and executed this webinar. So thank you, Jen. And with that, we will sign off, and everybody enjoy the rest of your day and your evening.
Take care.
Bye-bye.